Hello, my name is Tom McDougall. I'm from l'Université de Montréal, and I'm going to be presenting elucidating the automatically detected features used by deep neural networks for RFAM family classification. So a quick outline of what I'm going to talk about today. So the purpose and background of this work, the data, its preparation, and our implementation. Then I'll get into the results for binary and multi-class classifiers, and then I'll get into a little bit of a discussion on the sequence length and nucleotide composition and basically the features that these neural networks use in their classification performance and how much they require various features. And then I'll conclude. So purpose and background. Published results show that prediction of non-coding RNA family membership is achievable using machine learning systems. So using feature vectors that are generated from sequence alignments or using neural networks and convolutional neural networks. So in this work, we want to investigate if similar results can be achieved using deep neural networks trained on sequences directly. So we propose to verify that that's true, that if we can achieve that kind of performance, and also tackling the black box interpretability problem by selectively sort of hiding various features from the network and seeing how the performance is. So looking at the data in its preparation, we have over 7,000 RFAM sequences from 24 families, and then we prepared the sequences in a variety of different ways. So if we look at the top left figure, we can see that zero padding is an option that we can, because we have to pad out the sequences to a fixed length. So padding out to 600 nucleotides with zero padding, so just using a blank nucleotide that's in the nucleotide alphabet, or randomly padding, which is to the right of that. And then below we have shuffling the sequences, and then padding them either randomly or with zeros. And then we have shuffling the dinucleotide content of the sequences, which is what's over there on the right. On the far right, we have, for each sequence, shuffling the dinucleotide content. And I'll talk about how each of these preparation scenarios are important in a bit. And then for the target, for most binary classifications, one family remains positive, and then the other 23 are negative. And then for the dinucleotide shuffle, so again, all the way on the right, we have sort of shuffled sequences compared to real sequences, and then for multi-class, all classes have a unique label. So for the implementation, we have the length 600 sequence, and then we one-hot encode that using sort of a one-hot encoding of the nucleotide alphabet. And then from there, we feed it into one of two neural networks, so a bigger one, which is two hidden layers of 1,000 neurons each, or a smaller one, which is two hidden layers of 5 neurons each. And we wrote this using PyTorch and Python. So getting into the results, so for the binary classifier, so these values in the table are the area under the receiver operator characteristic curve. So we can see that in the most cases, it's very good performance for this binary classification. And we can see by the bolded results in those two columns that have bolded results. And we can see that they are performing quite well. I'll talk about that more in a bit, but before getting into multi-class results, which again, able to perform quite well. So we know that it's performing well. So we have a lot in the confusion matrix, we have a lot of correct results along the diagonal, and then also the accuracy is above 95% in these cases, depending on the architecture. So getting into more of a discussion, so where does this performance come from and what features are these networks focusing on? So if we look at the sequence length, so how much does sequence length affect the prediction ability? So we look at the zero padded case, where the sequences are padded with zeros, and then the random padding case, with the sequences are padded randomly. So with random padding, it's kind of like obscuring the length of the sequence, which would normally be very accessible to the neural network, where it is in the zero padded case. So with this sort of random padding case, we can see that on the right of the two red boxes, we can see that there is a loss in performance, but it's very, very small. So the network definitely doesn't need to be using sequence length to be able to make these predictions. Looking at sequence length and nucleotide composition, so if we look at the top figure, we see that the sequences are shuffled and then padded with zeros, versus just not shuffled and also padded with zeros. So when we shuffle the sequences, we're really getting rid of any dinucleotide information and also any sort of longer subsequence information. So that'll all be gone. We can see that there is a performance drop-off associated with that, and it's larger than the performance drop-off of removing only length. So it's interesting that we see such a rapid drop-off. And so we know that the network is garnering some of its performance from longer subsequences within the sequence. So moving into nucleotide composition, so we kind of combine two previous experiments here. We have the nucleotide shuffling with zero padding, and then also the random padding. So we combine that to nucleotide shuffling and random padding. So this removes, again, length from the padding, and then also nucleotide like subsequences and dinucleotide composition from the shuffling. So it really the network only has access to the nucleotide composition of the sequences, and we can see that the performance is it takes a hit, but it's the smallest of all the performances, the worst performing network. And we can see even when we up the size of the network to the big architecture, it still doesn't really reclaim a ton of performance, which is understandable because a lot of information has been removed from the data. Moving into a final case, so the dinucleotide content and longer subsequences. So for each real sequence, we have a corresponding dinucleotide shuffle sequence. So between the real class and the shuffled class, the length of all the sequences will be the same, their nucleotide composition will be the same, and their dinucleotide composition will be the same. And this is important because it means that the network has to focus on subsequences that are longer than 2 nucleotides to be able to make any classification. And we can see because the performance is better than random, it's capable of learning something based on that, which is interesting. So moving into multi-class results, so for RFAM families, we can see that the performance degradation kind of follows the same trends between the different experiments as we talked about in the binary case, with one notable difference, and that's that the small network is incapable of really learning anything. It's always poor performing, and it needs the much larger network to manage all of the different families. So in conclusion, we tested these different implementations of simple feed-forward neural networks in the context of RNA classification, using sequences as input with no structural processing step, and we use different shuffling schemes and padding schemes to figure out what features are being automatically detected by the network. And we got good results in the binary multi-class case. So we hope that this will reinforce the uses of DNNs for non-coding RNA annotation because we showed that they can capture these key features in primary structure and accurately predict families. And we want to be able to expand this DNN system to annotate non-coding RNAs across a whole genome. So picture like a sliding window across the genome and prediction for every few steps of the sliding window. And we want to compare that to state-of-the-art systems such as Infernal and Structurenator and others. So thank you very much.